Our next keynote speaker, Dr. Dieter Egli, Senior Research Fellow at the New York Stem Cell Foundation Laboratory. Now, of course, the New York Stem Cell Foundation we've honored in the past with the Stem Cell Action Award, and Susan Solomon is truly the founder of that organization, is a hero of the revolution. Imagine starting from scratch a foundation to support scientific research in stem cells from $0 to raising approximately $150 million and establishing world-class laboratories in just a few years. Her achievement and the achievement of her foundation, including Dr. Stephen Chang, who's their science director, is absolutely astonishing. And one of the stars in their constellation is our next speaker, Dieter Egli, who had the good fortune to meet in person in Vancouver at the ISSCR meeting. I got a chance to sit across from him at a New York Stem Cell Foundation reception. And we had the most engaging conversation because, of course, those of you who know the backstory of the Genetics Policy Institute and my own involvement, was very much involved with the area of SCNT, somatic cell nuclear transfer, also known as therapeutic cloning of stem cells. And the battle between something that we are all opposed of, reproductive cloning of babies, and research on stem cells, somatic cell nuclear transfer, there was not necessarily the bright yellow line dividing them. The world was quite confused about that. And of course, Genetics Policy Institute, my organization, organized a conference for the United Nations on that subject and really worked very hard so that those countries that wish to proceed with this with the proper ethical guidelines may do so. Well, New York is a progressive state and has facilitated this research. And Dieter Egli brought forth a dream that had run onto the shoals of scientific misconduct at one point, but he brought it home as one of the very few labs in the globe that is doing this work. Furthermore, the work with mitochondrial DNA and seeking an advanced way to solve problems with infertility, the very cutting edge work is being done by this wonderful young scientist, Dr. Egli. And he was the feature of a major spread story, I don't know if you saw it, in the New York Times Magazine. So he is a rising star of the field. His full bio is on your app. Please take a look at it, and I'm going to invite him up now, Dieter Egli, to provide one of our keynote addresses of the meeting.
Thank you very much, Bernie, for that introduction. It's great to be here in San Antonio. Can everybody hear me? All right. It looks like this is the pointer. Perfect. Yeah, so this is a field. Perhaps the presentation will come up soon. This is a field that has seen its ups and downs. It has had stellar moments, but it has had trouble sometimes. And I'm going to take you a little bit through that journey that now made it finally possible to generate cell lines that have stem cell lines that have the genotype of a patient and can then be differentiated into the affected cell type, and they're generated using human oocytes. So that dream started when John Gurdon in the 1950s was first able to clone frogs, and then in the 90s it became clear that it was also possible with mammalian oocytes. Oh, I see the presentation is up. Okay, that's good. Why is it not showing here? It can't. No?
It shows here so that you can see it.
All right, thank you. So, all right, sorry, I don't have my glasses on. I can't see my own presentation. So, all right, we saw that sheep were cloned. It worked with mammalian cells. And then, I'm sorry, this is embarrassing. Oh, yeah.
Would this be helpful to follow with the laptop?
Apologies. All right. So the concept of therapeutic cloning was after it was shown that it could be done with mammalian oocytes. Dolly, the sheep, is probably a term you're familiar with. It became clear that this could possibly be done with human cells. And so some remarkable scientists, among them Alan Coleman and Robert Lanza, they published the concept of therapeutic cloning. This was in the late 90s. And so what it entails is you have you take a biopsy from a patient, such as a diabetes patient, and then you perform nuclear transfer of these somatic cells into the eggs, and then you derive those stem cells. And these stem cells have the genotype of this patient. And so there is a dual use of those cells. They can be made into the differentiated cell type, shown here. And then you can either study that disease on the relevant cells, or you can use them for transplantation. And I think we are still realizing that dream. At that time, this was just a concept. None of them that had been realized. But it seemed very imminent. If it worked so well with animals, why wouldn't it just work with human cells? But many of you who work with human cells know that human cells very often behave a little different than animal cells. Things that work on animal cells don't always just work when you try to translate them to human cells. Equally, we know how to cure mice from many diseases, but we don't always know how this translates to a human organism. So there are some clear and significant differences between animal models and human cells. And so what was easy to do, of course, is to take skin cells and just put them in culture. We know that for a long time. But then the rest required a very significant effort. And because it involves human eggs, this was a very sensitive topic. It wasn't clear. Some people disagreed with that type of research because human eggs, they're germ cells. And for some reason, we discriminate between research on sperm cells, which also are germ cells. This is not as controversial, but human eggs seem to be a very controversial thing to do research on, despite this being one of the most important cell types and deserving study. And so, not too long after that therapeutic cloning concept was first proposed, some researchers started to do somatic cell nuclear transfer studies. And that immediately made some controversy, and it seemed it became necessary, and this is where Bernie Siegel had an important role to prevent a shutdown of that entire research at the level of the UN. That was even before I entered that field, and I had no knowledge until Bernie told me about that at ISSCR in Vancouver. And so it's remarkable the steps that this field had to go through until it finally became successful. So and then in South Korea there were, of course, very significant studies, but they all had failed, and I will get a little bit into it why probably they did not succeed. At the time when it turned out that the South Korean experiments had not succeeded, Harvard University decided that they would go forward with human somatic cell nuclear transfer studies. That made quite the news. At that time, we didn't know yet that the cell lines that had been generated in South Korea were not existing. And so, as we started to gear up for these studies, and that was then a postdoctoral fellow in Kevin Egan's lab, at Harvard University, also working with Douglas Melton. As we were starting these studies, rumors started to circulate that Shinya Yamanaka had succeeded in making human-induced, or first mouse, but induced pluripotent stem cell lines in 2006. And so in 2007, he also made human cell lines. And so you can see here that those ups and downs of the nuclear transfer field are tremendous. And I think it's unlike any other field that you in stem cells. So when Dolly was first cloned, there was a huge hype, and people thought, in the next few years there's going to be another you, and we know that's not the case. And then in 2007, as it was shown that stem cells can be derived in an alternative way using just 4 transcription factors, which seemed a much more simple way to do it. And so this led many researchers, including Jose Sibeli, who himself had worked on somatic cell nuclear transfer, to state that probably that therapeutic cloning approach is dead. It's no longer viable. This is not going to happen. But a few people persisted to get there, among them myself. And so it became possible for us to show in 2011 that human eggs can turn a skin cell into a stem cell. And what we did is transfer the skin cell into the egg and observe development and derive stem cells, and I will get back to the science about that. And then in 2013, the group of Shukrat Mitalipov showed that they could derive stem cells from diploid stem cells from fetal fibroblasts. So that was a remarkable achievement. And just a year later, my own group and another group surrounding Robert Lanza, they showed that they could derive stem cells from adult cells, and we showed that we could derive stem cells from a type 1 diabetic. So that was, in fact, quite a big part towards the realization of the therapeutic cloning. And you can see here, these are the studies that have come out so far. It's 4 studies, and this led the New York Times magazine and others to put this again on their covers. And so Jose Sibeli, who had earlier said that probably the therapeutic cloning is dead, he came back and said, well, it's actually doing quite well now. So what we did is we brought the field back on track through quite a long journey, and it was a quite remarkable approach. So why do we do this? That's, of course, now the important question. This was just kind of a historical background of how we got there. But what's most important is, of course, why would we do anything like that? Well, as I alluded to you earlier, somatic cell nuclear transfer could be a source of cells for cell replacement. We can make patient-specific cells now through that approach. In addition, there is reasons to better understand the human egg. How does the human egg turn a skin cell into a stem cell? How is that reprogramming happening? This is an approach that needs to be studied. It's scientifically very interesting. And then, of course, and what I'm going to talk about today as well, is it provides a point of reference for other reprogramming methods, including induced pluripotent stem cells. So here, a short overview of how somatic cell nuclear transfer works. Most of you might already know that, but now this is a human egg here, and you can take out the DNA here with a micromanipulation, and then what's done next is the fusion of a somatic cell, and so you end up with the egg that contains the somatic cell genome, and there is no fertilization involved. So the egg, the development of the egg is usually triggered by fertilization. That trigger involves a calcium pulse, and so instead of using sperm here, what we do is provide the calcium pulse, and that's called artificial activation. And then the egg can divide and develop, and hopefully it makes a stem cell line. And so in addition to just the logistical difficulties of getting that research started, it soon turned out that human eggs behaved very differently from stem cells, from eggs of animals. When we used protocols that were established on sheep or cows, they just did not work on human eggs. And that's also probably why the efforts in South Korea were not successful. That's why many other efforts that various groups had done over the years were not successful in deriving stem cell lines by human somatic cell nuclear transfer. So we had to start a very systematic approach to improve the development of these nuclear transfer cells. As you see here, in the beginning, we were successful in obtaining cleavage. So this is a cleaved egg that made about 8 cells, but it didn't develop any further. So what did we do? We knew that the reprogramming of somatic cells could be enhanced by the addition of histone deacetylation inhibitors. And so that was known from mouse somatic cell nuclear transfer studies. It was also known from studies on iPS reprogramming. So it made sense to add a histone deacetylation inhibitor after human somatic cell nuclear transfer. And now, for the first time, we were able to obtain these so-called blastocysts. Here you can see this is a hollow structure, and the outer layer here is a trifectoderm, and here this is the inner cell mass. And so what this means is that now we had the cells that could be used for stem cell derivation, because stem cells are derived from this little cluster of cells, the inner cell mass. And here you can see that we performed a very gradual optimization. So what this shows, this actually shows the percentage of eggs that develop beyond the cleavage stage, so where we previously found they had arrested. And you can see that with gradual optimizations, the addition of this HDAC inhibitor, which is the histone deacetylation inhibitor. I think the laser pointer doesn't quite reach there, but you can see that at the bottom, HDAC inhibitor. And we also made changes to the artificial activation. Instead of just the calcium pulse, we added 2 inhibitors, a translation inhibitor and the kinase inhibitor. And that now led to these bars here. That means there was development to the blastocyst stage. An additional modification that we did was we realized that the transfer of the somatic cell nucleus leads to calcium influx. And calcium is what activates the egg. And at the time of transfer, we didn't want that. We had to wait for the activation to occur until about 1 or 2 hours after transfer. And so what this green fluorescence here shows, this shows that there is calcium influx. This is a calcium indicator. And so we had to use calcium-free medium to prevent that influx of calcium. And that is the last bar on the side here. You can see here that development further increases. And some of the blastocysts we obtained were remarkably similar to an IVF blastocyst. They had a large and expanded trifectoderm and a distinct inner cell mass here in the middle. So what we did, and this is research that proceeded over several years, is we made several optimizations of the nuclear transfer protocol. One of them I mentioned is at the artificial activation, and that is in agreement with what the group of Shukrat-Mitalipov has done, is they also modified the artificial activation, and they found that this is a very important point to achieve development to the blastocyst stage. And then what we added is histone deacetylation inhibitor. That was an important point. And so these modifications, they allowed us to obtain, this is the third modification, no calcium here, the artificial activation and the histone deacetylation inhibitor. These modifications allowed us to obtain blastocysts at a reasonable rate and derive stem cell lines. And now this is a stem cell line that we derived from a type 1 diabetic subject. This person, a woman, had diabetes for many years, and you can see here that the cell lines we derived from these skin cells were diploid. They had 46 chromosomes. They expressed those typical pluripotency markers on the right, like OCT4, NANOG, and those we are well familiar with. And below, this is a characterization by microarray analysis. On the left is pluripotency genes are expressed. On the right is somatic genes are silenced. And so clearly these were fully reprogrammed somatic cells. And that was wonderful to see. And what was of course very relevant is the question of whether these could again differentiate into insulin-producing cells because they were derived from a type 1 diabetic. And so upon differentiation, they were able to show that these cells could differentiate into all cell types of all 3 germ layers, and here you can see some neurons, and here you can see the PDX1 positive cells and insulin producing cells. So this was upon differentiation into the pancreatic lineage, and these cells could secrete the insulin on the very right into the medium. So we had realized at least part of that circle that I showed you in the beginning that was initially proposed in the 90s. As you can see here, it took many years to do that, but it's wonderful to see that this was now successful. So what the conclusions from this part of my talk are, is we can now derive pluripotent stem cells by somatic cell nuclear transfer. And these nuclear transfer stem cell lines can differentiate into insulin-producing cells. So now we have induced pluripotent stem cells, and we have this method. And of course, induced pluripotent stem cells can do that too. They can also provide cells, we can do pluripotent stem cells from somatic cells, and it has also been shown that from type 1 diabetic subjects you can do that, and you can also differentiate them into insulin-producing cells. So what are the reasons to do this research? What can we learn from it? An important question that many people wanted to know about is whether perhaps these induced pluripotent stem cells are identical, or how close are they really to pluripotent stem cells derived from embryos. And so this is still a kind of a matter of debate today, because these comparisons are quite difficult to make in a human system. In the mouse, you have great functional assays for a pluripotent stem cell. You can make a new mouse, but you cannot do that in the human system. So we do not have good functional assays in human, and that's why we need very good reference points, and that's what the nuclear transfer system can provide. Nuclear transfer is, of course, very different in its approach. As I showed you, it uses the human egg, and it develops through those stages of pre-implantation development, cleavage stage, and then blastocysts, and then we drive the stem cells. So it follows a developmental path of reprogramming. While iPS cell generation is a very artificial process. There is ectopic expression, a forced expression of embryonic genes in the somatic cell, and we don't quite know what the consequences of this are. We essentially force cells into a state that does not exist anywhere but in the culture dish. It does not exist during human development. It's an artificial state. So could that have any consequences on the resulting cells? It seemed plausible that this could be. And in fact, there's a number of reports that suggested there are differences between ES cells and iPS cells. And among them, iPS cells apparently had a large number, or a moderate number, of new coding mutations. And this is a study that was published by Shinya Yamanaka's group that also caused some doubts on the integrity of iPS cells. And what they found is that differentiation defective phenotype, in that not all iPS cells differentiate well. Others have seen that iPS cells somehow seem to remember where they come from. So what somatic cell type they were derived from, whether it is from blood or from another source, like fibroblasts. And then others have found, in addition, that iPS cells sometimes lose imprinting. It means the uniparental expression of genes that are usually just expressed from one allele. And so this, of course, raises considerable questions whether these iPS cells are appropriate for applications like cell replacement, and whether what we should do about that, and whether nuclear transfer also has these problems or not. And more recently, a couple of studies were published that suggested that nuclear transfer cells might be superior in their reprogramming. And this study here was done in the mouse. These two were done in mice. And this one showed that there was difference in telomere elongation. This one suggested that there were less de novo coding mutations after somatic cell nuclear transfer. And this study suggested comparing fetal fibroblasts derived after nuclear transfer or derived after iPS method. This study suggested that the nuclear transfer was better at reprogramming somatic cells than the iPS method. So we wanted to know, how does this look in our cells? And what you see here is our experimental setup. We derived cell lines here by nuclear transfer in red from two somatic cell sources. This was from adult fibroblasts from the diabetic, ID 1018, and this from BJ fibroblasts. This is neonatal fibroblasts. And we also had, as a comparison, an ESL line derived from an IVF embryo. What we did is we performed gene expression analysis, imprinting, DNA methylation analysis, and coding mutations. And we looked at all these parameters. So what you see here is a cluster diagram of gene expression using RNA sequencing. And you can readily see that, so this is what we compared. And here is the cluster diagram of gene expression using RNA sequencing. Of course, all these cell lines were very different than the somatic cells on the right. But you can see here that the nuclear transfer cells and the iPS cells here cluster of the same genotype clustered closely together. Certainly there was no common clustering of iPS cells as opposed to nuclear transfer ES cells or the ES cells in general. So that suggested that both iPS cells and nuclear transfer ES cells generated cells that are very, very similar with regard to their gene expression pattern. What about DNA methylation, which is, of course, the basis for gene expression patterns? Here again, what we found is that the nuclear transfer cell line from the adults clustered with the corresponding iPS cell lines of the same genotype. And that was true for both the neonatal cells on the right and the adult cells on the left. And they also clustered equally close to the IVF ESL line. So again, clearly there was no common clustering of iPS cells as opposed to nuclear transfer cells. The genotype played a much larger role in the clustering than the method of derivation. So again, that meant that both methods resulted in very similar cell types with regard to DNA methylation patterns. What about the number of de novo coding mutations? So we sequenced the exome of, I apologize. So this is the DNA methylation patterns you can see here. This is gene expression. This is DNA methylation. And now here we have the number of de novo coding mutations. So we sequenced the exome of these stem cell lines, and both, again, the iPS cell lines and the nuclear transfer ESL lines. And we found that they both contain the exact same number on average of de novo coding mutations, 10.5 per cell line. So there was no difference. What about imprinting? We sequenced, because we had RNA sequencing results, we could look at the allele-specific expression. So imprinted genes, shown here for age 19. What you see here is if there is an A, a large A, that means there is a monoallelic expression. And for these two cell lines, number 5 and number 6, there's clearly just one allele is expressed. For that cell line 8, there are both alleles, so there's biallelic expression, the A and the G, and that's not normal. And you can see here that because this gene had a number of SNPs, this biallelic expression did hold true throughout the gene, and for the other two cell lines, there was monoallelic expression throughout the gene. And if we counted the number of genes that contain such abnormalities, and also looked at the DNA methylation pattern, we again found that nuclear transfer ESL lines and iPSL lines behaved identical. They had below here, they had a similar number of these imprinting aberrations, both iPSL lines and nuclear transfer ESL lines. On the left, at the bottom in green, you see ESL lines. These also have a low number, but clearly a significantly lower number of such imprinting aberrations, which probably occurs due to extended cell culture. Up here, what you see here is that the DNA methylation levels at the imprinted genes. And normally, in such imprinted genes, there should be about 50% DNA methylation. One allele is methylated, the other one is not. But if there is demethylation, like in here, in this cell line, there is also biallelic expression, indicated in B at the top. So the DNA methylation patterns agreed with the expression of both alleles. So we knew that these imprint aberrations are real. But they did not occur in all cell lines, and they did not always occur at the same locus. So what we can conclude from these studies is there is no clear winner when comparing the two cell approaches to reprogram. With regard to the parameters we looked at, they result in highly similar cell types, gene expression, and DNA methylation patterns. Both nuclear transfer and ESL lines show differences to ESLs derived from IVF embryos, and that is with regards to the number of de novo mutations and imprinting aberrations. And so that most likely means that for both methods, if such cells will be used for applications such as cell replacement, the appropriate quality controls will be needed. With this, I would like to switch to the second topic. So far, I have shown you that we can use human eggs to reprogram somatic cells to a pluripotent state. That could be useful certainly useful for research, and it could also be useful to develop cell replacement therapies. So what can we do with the human eggs and nuclear transfer with regard to reproduction? And this is one opportunity, is to use such techniques to prevent the transmission of mitochondrial disease. What is mitochondrial disease? Well, here you see one family is being observed and followed at Columbia University by the group of Dr. Michio Hirano. And what you see here is that mitochondrial disease is maternally inherited. This person here on the left is a father, and the children don't have any of that phenotype. There's a mild phenotype in this person. This person here is the mother. She doesn't have a phenotype, and she transmits the mutation, the mitochondrial mutation, and the disease to her children. Here again, you see in this family tree, circles women, squares men. You can see again on the left, this father here on the left didn't transmit the disease or the mutation, was affected. But on the right, the woman has 4 affected children, and one of them died. So these can be very severe diseases. They can often be lethal, and they're obviously a huge burden for families. They're caused by mutations in the mitochondrial DNA. And even though the mitochondrial DNA is very small, it's just about 16 kb, it has about 32 genes, it can have a significant number of mutations that cause severe disease. Unique about mitochondria is that they are not inherited in a Mendelian way. So we cannot use pre-implantation genetic diagnosis the way we use it for like cystic fibrosis or sickle cell anemia for the inheritance of these nuclear mutations. But what can be done? What we know is that these mitochondria, there are many mitochondria within one cell, and so there are also many copies of the mitochondrial DNA. And so it depends how many copies of that mitochondrial DNA is mutated, whether a person will be affected by the disease or not. So the disease severity is dependent on the affected genes and the percentage of normal mitochondria versus mutant mitochondria. There are many organs that can be affected because mitochondria generate the energy, the ATP of the cell. This is of course relevant for essentially any organ, but the skeletal muscle is of course very susceptible to that, the brain, and many other organ systems. And it's easily understandable that if mitochondria are severely affected, there can be a lethal disorder. So what could we do to prevent the occurrence of such mutations and to free families from the burden of this disease, while they still have their own genetically related children? So on the left, you see eggs from a woman who has one particular mitochondrial genotype. That could be the genotype that is affected by such a mitochondrial DNA mutation. And on the right, you see an egg that's healthy. And that could be an egg from an oocyte donor, an anonymous or a related donor who doesn't have that mitochondrial mutation. What we do is we can remove the nuclear genome from this egg, just as I showed you for the somatic cell nuclear transfer. Remove it also from this egg. This nucleus gets discarded, and this nucleus is transferred into the healthy mitochondrial environment. And what we did to test that, the feasibility of this, is we used parthenogenesis. And there were both scientific as well as practical reasons to do that. In parthenogenesis, we don't generate any viable embryos, but we can still test the development and the effect of this manipulation on cells in vitro. Because parthenogenetic cells, parthenogenetically activated oocytes, that means there's no sperm involved, they can develop to the blastocyst stage and they can give rise to stem cell lines. Also, an important scientific point is that if we do that manipulation on just eggs without fertilization, there is no way a normal sperm genome can complement for the possible defect that we might be introducing during that manipulation. So we performed that exchange, just as I showed you in the diagram, and observed decent development to the blastocyst stage. You can see that some of these blastocysts are of very good quality. For example, this one, large refractoderm and a nice inner cell mass. So that was very encouraging. But of course, the question is how much of those mitochondria are carried over from one egg to the other? Because the nuclear genome is condensed and it's associated with microtubules, the so-called spindle of the metaphase 2 egg, shown here on the left. The red are the mitochondria. You can see here that the genome is essentially mitochondria-free. The genome is removed in a small vesicle of cytoplasm, and so a small amount of mitochondria are indeed carried over, shown here on the right. But we can quantify that because the two eggs from the two different women had different mitochondrial genotypes. We can quantify how much of the mitochondria is carried over. And desirable is, of course, as little as possible, because if there is a mutation among those mitochondria, we don't want it to be carried over. So what we found is that initially, these are various stages after the transfer. So this is at the cleavage stage, shortly after egg activation, shortly after transfer. This is the molar and blastocyst stage. And then here, on the right, these are stem cell lines that were derived after that transfer. You can see here that less than 0.5% of the total mitochondria contained in that egg originate from the original egg. So less than 0.5% are carried over into the new egg. And that's, of course, a very low percentage. That's very good. What was even more encouraging to see is that on the right, you see that these carried over mitochondria, they disappear. They are lost, and the majority genotype of mitochondria takes over. So in stem cell lines derived after this approach, and we did anything one could do to those stem cell lines, we grew them for 1 year, we differentiated them, we reprogrammed them back again, and we never found that the original mitochondrial genotype would re-emerge. It remained a pure new mitochondrial genotype. And so that showed that we could essentially replace entirely the mitochondrial genotype using this manipulation. And that would, of course, also mean that we would replace any mutation associated with mitochondrial disease. A chief criticism of this approach by some is that, well, now you make a new combination of nuclear alleles with different mitochondrial alleles. So could that have any effect on mitochondrial function? Well, needless to say, every fertilization event makes a new combination of nuclear alleles with mitochondrial alleles. That's why we are all different. So this is not unique to this approach. This is simply something that's inherent to human reproduction. But nevertheless, we wanted to address that question. And so as it occurred is that the two mitochondrial genotypes, they had alleles in genes that coded genes. So here in the tRNA, leucine, there was a polymorphism. And here in this gene. But chiefly, this was a polymorphism that actually altered the sequence of the tRNA, and in principle could have a functional effect after doing that exchange. And so what we did is we had stem cell lines and differentiated those into skin cell lines and compared them to the skin cells of the oocyte donor. So the stem cell derived skin cells on the right had the same nuclear genotype as the skin cells from the skin of the egg donor, but now had a different mitochondrial genotype. And so we could ask the question whether mitochondrial function was normal. And that's shown here. So on the right, you see, so these are the original skin cells in blue and red, and these are now the exchanged stem cell derived skin cells that have the nuclear genotype of the red cell line, but the mitochondrial genotype of the blue cell line. So there are the nuclear alleles are combined with a new mitochondrial genotype. But you can see they behave just the same as this nuclear genotype in the red collaborates with the blue mitochondrial genotype just as well as the original cell line on the very left. So clearly the exchange had no effect or did not compromise mitochondrial function at all. We would expect that, but it was important to show because this concern was mentioned. So with this, I would like to wrap up and say these are the take-home points that I'd like you to remember. We can use somatic cell nuclear transfer for the regeneration of stem cells, for example, for diabetes. And also nobody, to my knowledge, is seriously considering reproductive use of that technique. It wouldn't be advisable. It's not because, as we showed in our studies, there are a significant increase of de novo coding mutations, and there are imprint aberrations, which would definitely result in severely abnormal development. These cells, derived by somatic cell nuclear transfer, could, however, be extremely useful for incurable diseases such as diabetes, to generate replacement cells. Then genome transfer between oocytes. The second part of my talk is we showed that this could be effective to prevent the transmission of mitochondrial disorders, and this could be useful for reproductive use. Now the two very often get confused simply because they both use a needle and a method that involves a microscope and a micromanipulator and involves a nucleus. It's sometimes hard to differentiate the two. This cannot be put into the same box. A somatic cell that is transferred into an egg is not prepared to undergo full-term development. This is not the way nature or is designed, or the way development normally occurs, I should say. In the second part, this is the normal trajectory of a germ cell genome. All we do is transfer the genome of one egg into a different egg. So that genome is prepared to undergo full-term development. So it is here, this is appropriate for reproductive use because we're using a germ cell genome. This one is not, and that's also why it's associated with these defects. So with this, I would like to thank the many people involved in this research. Most of the nuclear transfer studies, in fact, all of them have been done at the New York Stem Cell Foundation laboratory. This was only possible because Susan Solomon and Kevin Egli and others, they stepped forward and said, we need to do something about this, and started a privately funded lab in Upper Manhattan. And very important was the work of Dr. Mark Sauer, who started the egg donation program for research. This is a very unique program, that one of its kind, that allowed to perform these exciting studies. Also, Shukrat Mitalipov from the University of Oregon, he contributed DNA for the analysis of nuclear transfer cell lines and mutation rates. And then we had a wonderful collaboration with Nissim Ben-Venisti of the Hebrew University of Jerusalem on comparisons of nuclear transfer cells and ES cells. And with this, I'd also like to point out that, in a few hours, there will be a small panel on discussing more about how did it become possible to get eggs, what are the ethical implications of this, what are the regulations behind it. I haven't been able to go into that at all, but for now you have a background on the science. And thank you. Any questions?
Yes, you can ask a question.
Of course, you know I really like that work, but the thing I wanted to ask about the paper that just came out, I know that Egli and English on it.
Yes.
I think that study in which they compared clones and essentially they were looking for the, thank you. They were looking for the effects of mitochondrial mismatch with the nucleus on transplantation of mouse nuclear transfer cells. And they found that the mitochondria caused rejection of the cells. And I was wondering if you've thought through the implications of that for humans, or if you expect something to be different about humans so that that would not be the case.
Well, it's a very useful study. It certainly needs to be incorporated in when considering making cell lines using somatic cell nuclear transfer for cell replacement. It's probably better to have a mitochondrial match, or at least not a significant mismatch, in terms of are there any coding changes in the mitochondrial DNA that might elicit such a immune response. My understanding is still that the immune response is rather minor. And I think the authors in that paper also mentioned that they mostly used undifferentiated ES cells for these assays and not differentiated cells, and they mentioned that this could play a role in the immunity that they see. So it's not 100% clear whether the mitochondrial genotype is solely responsible for that effect.
So I probably haven't read all the way through the end of the paper either, but I did see something about just transplanting mitochondria and getting an antigenic response.
Yeah, I think what will need to be done for these studies when generating somatic cell nuclear transfer cell lines for transplantation, one will need to match mitochondrial DNA to some extent to avoid that from happening.
Right. So thank you very much. Our speaker will take some questions on the side because we have to keep on schedule. Dieter, thank you so much. Let's give a round of applause.